In this lecture, we focus on the statistical foundations of molecular QTL mapping, starting from the linear model for eQTLs and building toward single-cell data in the next lectures. eQTL mapping tests whether a genetic variant is associated with the expression level of a gene across individuals, as we plot here with 3 different genotype groups for the SNP on the gene expression. A variant that decreases or increases gene expression with the increase in the number of effect allele from 0, 1 to 2 is called an eQTL. eQTLs are the most widely studied molecular QTL type, making them the foundation for interpreting GWAS associations. With that in mind, let us look at the types of molecular QTLs and the statistical models used to map them. Gene expression is one of the several molecular layers we can map genetic effects to. This table summarizes the major QTL types, all measured from bulk sequencing assays. The top 4 rows all use linear regression on continuous, normalized measurements. These phenotypes, gene expression levels, protein abundance, chromatin accessibility, and histone marks can take any positive value and are well behaved after normalization. So the standard linear model applies directly. However, the bottom 2 rows are quite different. Methylation is measured as a beta value, which is the proportion of DNA molecules that are methylated at a given CpG site, which is bounded strictly between 0 and 1. And splicing is measured as a ratio of reads supporting a particular intron excision, also bounded and compositional. Plugging these bounded values into a standard linear regression can be problematic, so different statistical models, as listed here in the table, are needed for these 2 types of QTL mapping. Despite these differences in statistical model form, the core statistical question is the same for every row in this table, which is genotype of a variant associated with the molecular phenotype across individuals. In this lecture, we will use eQTL mapping and its linear regression framework as the worked example, and the logic transfers directly to the other QTL types. So eQTL mapping is based on linear regression, and normalized expression Y is modeled as a function of genotype G, coded as 0, 1, or 2 for the number of effect alleles, plus covariate C and the residual noise. The parameter we care about is beta 1, the change in expression per additional allele. If it is significantly different from 0, we have an eQTL. In practice, we focus on cis-eQTLs, where variants tested within 1 megabase window around each gene, since most regulatory variants act close to their target. But the same linear model also applies to trans-eQTLs, where the variant and the gene are far apart or on different chromosomes entirely, though trans analysis can require much stricter significance thresholds because the number of tests are much larger here. And the model relies on 2 key assumptions. First, expression Y is approximately normally distributed. This can be satisfied after log normalizing or inverse normalizing bulk RNA-seq data. Second, observations are independent, suggesting each individual contributes 1 data point and there is no correlation between samples. Both assumptions can hold generally for bulk data. But as we all will see in the next lectures, these assumptions can break down in single cell data, which motivates us to use new class of models for EQTL mapping in single cell data. And then let us now see what this test looks like in the data set. So first, let's visualize EQTLs. We can split individuals by genotype, 0, 1, or 2 copies of the effect allele, and plot expression in each group. And slope here is represented by beta 1 in the linear model, shows the rise in mean expression per additional allele. And this plot actually appears in every EQTL browser because if the three distributions overlap at the same level, there is no effect as beta 1 is close to 0. So if they separate cleanly with a linear trend of increase or decrease, we can say the signal is strong. So for each gene variant pair, we can estimate beta 1 and its standard error and the p-value based on statistical tests and based on this linear regression model. And we can use t-test, f-test that can help suggest if beta 1 is significantly different from 0 or not. And then let's take a look at how we handle the practical considerations based on this model in EQTL mapping. First, for covariates, and we generally include genotype principal components to control for population structure, plus age, sex, and batches as covariates. And we usually add peer factors, which are latent variables that help account for unmeasured confounders, like cell type composition, which can help substantially boost discovery. Second, we need to consider multiple testing because we're running millions of variant gene tests. Within each cis window for a gene, and variants can be in LD, and the permutations are usually used to get a gene level p-value. And then we apply FDR correction across genes. A gene that passes the threshold is called an E gene, and its top associated variant is its lead variant. And GTAGS has successfully applied this framework across 49 tissues from nearly 1,000 donors and identified approximately 16,000 E genes. So one of the key insights from bulk EQTL studies is that genetic effects on gene expression can often be tissue specific. For example, a variant may regulate a gene's expression in liver, but not in blood, lung, or thyroid. Identifying these tissue-specific EQTLs has been essential for connecting GWAS loci to the tissue context where they act. But within a single tissue, cells can be quite different. They can differ in cell type, cell state, goes through different cell cycle and developmental and metabolic stages. And the limitation of bulk RNA-seq is that it can miss these cellular variations by measuring gene expression from the entire tissue. And capturing this biology requires measuring gene expression at the level of individual cells. And that is exactly where our next lectures pick up. And we'll see statistical models needed to conduct EQTL mapping using single cell data. So this very last slide just lists the references for those important resources that we include in the table for different molecular QTLs.